To conclude the overview of the genetic work in neuro-oncology, Samantha Mascelli will explain how the initially huge amount of data which was produced by the gene expression analysis could be reduced to a much more manageable number thanks to a special chip developed for Healthy Child by ASPR, Healthy Child's Estonian partner, and how this has led to some interesting results.
So we perform a gene expression analysis on those cases, starting from microarray technology that furnish us a huge amount of data, a huge number of genes that have need to be cleaned in order to reach a smaller number of genes that we have validated with the real-time PCR DISI group. And we got 140 genes differentially expressed on low-grade glioma, considering the query, the site of the lesion, so cerebrum versus cerebellum. And specifically for the pilocytic astrocytoma, 43 genes differentially expressed emerged from the analysis of the microarray data. And so a specific genetic signature related to tumor site emerged, and the genes implicated in this topic for this signature codefined for cell cycle communication, focal adhesion, cell differentiation, and development. And a list of 18 genes that represent the most statistically significant genes implied in the low-grade glioma were selected with these statistical methods. And different genes as FOXG1, ARX, LHX2 are homeobox genes that are implicated in neural development and in specific in regional brain morphogenesis. Other genes implicated are transcription factor or other genes implicating the progression in the cell proliferation. Here in this slide, the genes are organized upon the pathway where they are involved into. As you can see again, several genes are implicated, involved in anatomical structure morphogenesis, but we also found genes implicated in the cell proliferation, and there are several genes, 13 genes, implicated in the regulation of biological process. So this selection of genes were then validated with the real-time PCR, and we can assess a differential expression level of the genes in low-grade glioma. So our analysis showed a distinctive subset of gene expression profile among low-grade glioma that could represent possible genetic markers for these entities. Moreover, we highlighted a specific gene expression pattern for pilocytic astrocytoma, which is peculiarly related to the brain site. And as I already said before, we found different genes, homeobox genes or transcription factor that are aberrantly expressed in the glioma.